friends, welcome to Knowledge Center. I am Saknik Chakravarti with you all, and today I am going to start with ICSE Class 10 Biology. So, before going to each and every chapters, I will first recommend you all to take up your concise biology book with a pencil or a pen in your hand to underline or mark all the important points that I say. So, please open up to your contents page or index page. It is seen that the first chapter is cell, the structural and functional unit of life. So this chapter, as per your ICSE syllabus, it is not there in your syllabus, or any questions will not come in your ICSE board examinations from this chapter. So you are free to skip this chapter, but I will say one thing: it is not included in class 10 syllabus. It's okay, but in your future, in your near future, this chapter is the base of all biology. Cell is the primary source, or it is the very important but thus very micro source of biology, and the base, the most important thing, the base of each and every biology: animal biology, plant biology, human biology. That's why it is said the structural and functional unit of life. Okay, that's why it is said structural and functional unit of life. So it is, you are free to skip up this chapter for class 10 board examinations, but in class 11 and 12 this chapter is the most important. First of all, teachers will explain or teach you all this chapter about cell and discovery of cells and each and everything about it. So as I will look forward to your ICSE class 10 board examinations, I will also skip this chapter and I will go to the second chapter, that is structure of chromosomes, cell cycle and cell division. So let's take up this chapter. Okay, chapter number 2, structure of chromosomes, cell cycle and cell division. Before going to any of the topics, I will say you that this chapter I am going to do in two major parts. First part will comprise of structure of chromosomes and second part will comprise of cell cycle and cell division. So as it is the part 1 of this video, let's start with the first part of this chapter, that is structure of chromosomes. So before going to core concept of the structure of chromosomes, you should know what do the word chromosome mean. The word chromosome mean colored bodies. So if you want it to be written down, please write. It is in your book, but if you want that, yes, it is very important to be written down, please write it. Chromosome means colored bodies. Now we will go to the next part of this structure of chromosomes, that is the parts, okay, the constituents of chromatin material. What are the constituents of chromatin material that you should know? So the first constituent of chromatin material is DNA. Okay, so what is DNA? The full form of DNA is a very important thing that you should know, because many times the question has come that expand the following. First is DNA, so you have to write the full form of DNA. So it is written deoxyribonucleic acid. DNA's full form is deoxyribonucleic acid. And second part is histones. What are histones? The core knowledge of histones are not written in the book, or it is not in the syllabus. You have to just know only a thing, that histones is a type of a protein. Histones are type of a protein. So these two are the main constituents of chromatin material. Okay, now we will move on to molecular structure of DNA. Now it's very important to know what is the molecular structure of DNA. So each DNA strand means there are two DNA strands. So in each DNA strands there are cluster of nucleotides. So cluster of nucleotides forms DNA strands. So what are the components of now nucleotides? That is very important. First is a phosphate group, second is a sugar, third is a type of a base. So think this one, this L-shaped thing is a nucleotide. Now where do each of the constituents are present, that I will say you. In the top part a phosphate group is present, in the middle part a type of a sugar is present, and in the lower part what is present, a type of base. So phosphate group, sugar and a base. Now as I've said, a type of a sugar and a type of a base, you have to know which type of sugar and which type of a base are the constituents of a nucleotide. Sugar, which type of a sugar? It is pentose sugar. Please write, it is pentose sugar. And which type of a base? It is nitrogenous base. Okay, it's nitrogenous base. So this is one nucleotide. Okay, similarly you think another nucleotide is here, phosphate, pentose sugar, nitrogenous base. Now what happens when, how do the double helix structure of DNA is formed, that you have to know. The double strand structure of DNA is formed, that you have to know. So there is two nucleotides, they get joined. Where they are getting joined? See, here is there is a nitrogenous base, here there is nitrogenous base, they are getting joined like this. Okay, so among the nitrogenous base there should be some bond, that is why they are getting joined. Okay, so that you have to know. First of all, you have to know the 4 types of nitrogenous bases that is included in your syllabus. First is adenine, second is guanine, 3 is cytosine, and 4th is thymine. Okay, you can write in any order, but a simple order is ATGC. A for adenine, T for thymine, G for guanine, and C for cytosine. Okay, ATGC. So 4 types of nitrogenous base you have to know, it's very important. Now among which two nitrogenous base a bond gets formed, that you have to know. As I have said, AT, that's why thymine bonds with adenine, thymine bonds with adenine, and cytosine bonds with guanine. Okay, now there are 2 types of bond here, double bond and triple bond. What type of bond? It's not ionic, not covalent, it's hydrogen bond. Okay, double bond and triple bond, 2 types of bond, double bond and triple bond, but the main bond that gets formed between both of the nitrogenous bases is hydrogen bonds, the weakest bond. You will study in your higher classes. Now between thymine and adenine, how many bonds gets formed, that you have to know. Double hydrogen bonds get formed. Okay, double hydrogen bonds. And in between guanine and cytosine, triple hydrogen bonds are formed. Okay, so this concept may come in your objective type of question in section A. Okay, between which nitrogenous base double bond or triple bond is formed, so you have to know. So like this, the 2 nucleotides like this all are formed, and the cluster of nucleotides form a DNA. Okay, now you have to know something about nucleosome. You have to know something about nucleosome, it's important. Okay, nucleosome, what is nucleosome, that I will explain you. A DNA strand, a single DNA strand. Okay, there are 8 histone proteins. Histone proteins is in a spherical shape, so there are 8 histone proteins like this. A single DNA strand winds up the 8 histone proteins. Okay, when the single DNA strand winds up the 8 histone proteins, the cluster of the collectively they are known as nucleosome. clear? When a single DNA strand winds up the 8 histone proteins, collectively it is known as nucleosome. Now I will say you the whole process of the structure of chromosome. When a single DNA strand winds up the 8 histone proteins, it coils up. Okay, it starts coiling, and it coils in a such way that it moves on to the phase of supercoiling. Okay, it gets supercoiled. You have seen the wear of a land phone. It is in a form of just a simple coil, but if you coil it more, it will move on to the phase of supercoil. Okay, it will be supercoiled. So like that, the one single DNA strand coils the 8 histone proteins. So like this, coiling, they forms a single chromatid. Okay, now you have seen there is a general tendency to see the chromosome in form of a crisscross shape, like this. Okay, we see a chromosome in a crisscross shape, but it's wrong to think like that. The first part, like this. Okay, as we see like this, the first triangular part is a single chromatid. First triangular part is a single chromatid, and second, the lower triangular part is a single chromatid. Okay, that you will know during the cell division part. I will say you. So first triangular part is a single chromatid, second triangular part is a single chromatid. So both of the chromatids gets joined to form a single chromosome. Both of the chromatids get joined. Now think, this is a chromosome, this is a chromatid, single chromatid, and this is a chromatid. Okay, this is a chromatid. So both of them gets joined here. So this point where it is getting joined, okay, both of the single chromatids where they are getting joined, this point will be known as centromere. Okay, where both of the chromatids get joined, it will, the point of their joining will be known as centromere. It is written in your book. Okay, please, if you want that it would be written, please write it. Okay, so like this we have came to the end of the structure of chromosome. It is clearly explained to you all. This is the first part of this chapter. So any doubt regarding the structure of chromosomes, please write in the comment section, and please be updated, because I will be posting the next part of this chapter, that will comprise of cell cycle and cell division. Okay, so thank you very much for watching.